Good morning, everyone. Today I would like to talk about the mechanism of the catalysis of chorismate to prephenate by chorismate mutase. Chorismate mutase is a central enzyme in the shikimate pathway. Shikimate pathway is an aromatic biosynthetic pathway that converts primary metabolites phosphoenol pyruvate and erythrophosphate phosphate to chorismate. The pathway is subsequently responsible for the production of the phenylalanine and tyrosine through the conversion of chorismate to prephenate. The diagram in the slide shows the entire shikimate pathway, from phosphoenol pyruvate and erythrophosphate phosphate conversion to shikimate conversion to prephenate, and then subsequently leading to the phenylalanine and tyrosine formation. Chorismate mutase catalyzes the Claisen rearrangement of chorismate to prephenate, which is the first committed step in the biosynthesis of phenylalanine and tyrosine. Shikimate pathway occurs in higher plants, bacteria, fungi, but not in animals and humans. This makes phenylalanine and tyrosine the essential amino acids that humans and animals need to obtain through their diets. However, tryptophan can be synthesized with the organisms who can hydroxylate phenylalanine, whereas the organisms who cannot need their tryptophan also through their diets. This also makes it an attractive target for the development of antibiotics or herbicides. Indeed, inhibitors of enzymes in the shikimate pathway have commercial applications. For example, phosphonomethylglycine, a potent inhibitor of 5-enol-pyruvyl shikimate 3-phosphate synthase, is a broad-spectrum herbicide which achieves more than $1 billion in sales per year. Chorismate mutase achieves its purpose of converting chorismate to prephenate through Claisen rearrangement. Claisen rearrangement is an important C-C bond forming reaction in organic synthesis. In its simplest form, it involves thermal sigmatropic rearrangement of allyl vinyl ether to 4-pentanal. It's a paracyclic reaction where a CC sigma bond, a CC pi bond, and a CO pi bond are formed, and two CC pi bonds and CO sigma bond is broken. When we compare the energies of the bonds that break with the bonds that form, we see that we are trading a relatively weak carbon-carbon bonds, pi bonds, about 65 kilocals per mole, with for a relatively strong carbon-oxygen pi bond for about 85 kilocals per mole, for a gain of about 20 kilocals per mole, which means this reaction is an irreversible reaction in nature. Now, the structures of the chorismate mutase enzymes. Chorismate mutase are unregulated homotrimeric enzymes that form a pseudo-alpha-beta barrel with 3 identical active sites situated in the interface region between 2 subunits. They can be allosterically regulated or not depending on the organisms they are found in. As you can see in the slides, the chorismate mutase one subunit of Bacillus subtilis and E. coli. They have entirely alpha helical structures. And as you can see, they're found as 2 subunits together in a dimer. And on the left side is the dimer of the Bacillus subtilis chorismate mutases. Picture in the slide depicts the active site of the chorismate mutase enzyme and the residues of the chorismate interacting with the chorismate substrate. The active site of the chorismate mutase from the bacteria Bacillus subtilis is formed by arginine 7, glutamine 78, arginine 90, tyrosine 108, and leucine 115. From one subunit, and phenylalanine 57, lysine 60, valine 73, threonine 74, and cysteine 75 from the other subunit. The importance of arginine 63 is supported by more recent crystal structure and computational studies and strict conservation of the residue in chorismate mutase sequences. Arginine 63 also makes contact with the ring carboxylate of the complex molecule in some of the 12 active sites present in one asymmetric unit. Arginine 90 plays a central role in the binding and catalysis. It is part of a clamp-like structure that orients the side chain of the chorismate with respect to its cyclohexadiene ring. The hydroxyl group of chorismate forms a hydrogen bond to the sulfahedral group of the cysteine 75, particularly to the sulfur of the cysteine, and to the carboxylate of glutamate 78, which in turn forms a salt bridge with the guanidinium group, which is a C-double bonded NH group of the arginine 90. The latter also makes hydrogen bonds to the ether oxygen and the carboxylate of the enol pyruvate side chain of the substrate. The carboxylate is further fixed in place by the side chain of arginine 7. Hydrophobic interactions with the cyclohexadienyl ring of the substrate are made by phenylalanine 57. The recruitment of these residues to the active site is one indication of dynamic changes of the enzyme upon substrate binding and catalysis. The active site thus exhibits the excellent charge complementary to the substrate and provides numerous hydrophobic, hydrogen bonding, and ionic groups for the molecular interaction and recognition. The removal of Q residue arginine 90 from the chorismate mutase enzyme destroys the balance of the interactions in the active site required for stabilizing the conformation of the transition state in the Claisen rearrangement of chorismate to prephenate. As seen in the picture below from the article by Dr. Hongguo and others, the arginine 90 deletion destroys the activity of the chorismate mutase. The article also reveals that the conserved substitution of lysine 90 in place of arginine 90 also destroys the activity of the chorismate mutase, showing the strict conservatism in the chorismate mutase enzyme. The formation of prephenate during the Claisen rearrangement, the carboxylate oxygen of the prephenate is not associated with any residues of the chorismate mutase. The tertiary carboxylate groups of the bound prephenate are relatively solvent exposed at the entrance of the pocket of the chorismate mutase. Potentially stabilizing contacts with the side chains of the arginine 63 and arginine 116 could be seen as seen in the picture below. The other studies, majorly conducted by Haar and Bruce, attribute chorismate mutase catalytic effect mainly to the generation of near-attack conformer of the substrate in the active site of the enzyme. This near-attack conformer is defined by a short van der Waals contact distance of 3.7 angstroms between the reacting centers in a conformation that is almost never adopted in water. The enzyme was shaped by evolutionary forces to accommodate the substrate structure in reactive chair-like pseudodiaxial conformation by providing appropriate charge complementary and hydrogen bonding interactions, and to stabilize the polarized transition state of the Claisen rearrangement. The enzymatic reaction, like its uncatalyzed counterpart, proceeds via a pseudodiaxial, chair-like pericyclic transition state, in which the transition state is stabilized by the residues of the arginines, leucines, isoleucines, valine of the chorismate mutase as described in the presentation before. The slide shows the overall Claisen rearrangement reaction of chorismate to prephenate, and this clearly shows which bonds are being broken and which bonds are being formed during the Claisen rearrangement. C1 and C2 pi bonds, C5, C6 pi bonds, and C4 oxygen sigma bonds are being broken, and C1, C6 sigma bond, and C4, C5 pi bond, and C2 oxygen pi bond is being formed during the Claisen rearrangement of chorismate to prephenate by chorismate mutase in this mechanism. Overall, though, this discussion of chorismate mutase has shown that the chorismate mutase has been a subject of extensive experimental and theoretical investigations. There are still some uncertainties concerning how Claisen rearrangement from chorismate to prephenate is actually catalyzed. Since chorismate mutase is very unique, as it is the only known enzyme to catalyze the pericyclic reaction in the transition state of the Claisen rearrangement, this unique enzyme will no doubt bring about many new and insightful research. Although the most likely mechanism of chorismate mutase have been suggested, more modern techniques are necessary to identify the definitive mechanisms of the catalyst. Overall, the information presented on chorismate mutase gives insight into the regulatory mechanism of the shikimate pathway and amino acid biosynthesis. Thank you. These are my works cited for the presentation.